Okay, morning everyone. So what I will talk about is AI and drug discovery, state of the art we will get back to as well, just in a slightly different way maybe than expected. So I will talk about AI and drug discovery and how difficult it is to translate models into practice. It's easy to train a model, right? But the question is, if you have a machine learning model and you work in a pharma company, can be big pharma, can be a small one, the question is, how do you then change a process, right? How do you change what people do? Is the data that we have actually suitable for what we want to achieve in practice, and so on? And 4 or 5 years ago, I started thinking about the questions we're trying to answer with machine learning and drug discovery, and then to consider which data do we actually have, how do we use that data, and can we actually achieve what we aim to achieve? And I think there's a gap, basically, between those two things. I will guide you through that a little bit. And so these are the 6 circles we'll talk about. So drug discovery means putting compounds into patients, into humans. And you want those compounds to be efficacious, to achieve their goal, and you want to mitigate safety. So you don't have compounds that are safe in absolute terms, but you want to have tolerable safety. Okay, you want to achieve your endpoint, safety, and you want to have acceptable safety, the endpoint efficacy, and you want to have acceptable safety as well. Okay, that's at the goal if you want to do drug discovery. And of course, you might also realize if you do machine learning, usually you don't have compounds you put into the clinic, right? You train a model and then you have some cross-validation or test set, right? And you're actually quite far away from that. And I will explain how those two, the short-term measurables and the long-term goal, how that actually relates to each other. But the goal is actually clinical in drug discovery. You want to have a compound in patients, safe and efficacious. But there are those 6 circles of hell that prevent us from doing that in many cases. I will guide you through that. And that's biological data that is different from data in many other disciplines. It's difficult to label life science data. That's a very short version. We'll come back to that more extensively later. Then there's the problem how we measure model metrics. And it's quite easy to lie into your own pocket and to say, I have a perfect model, right? What matters is the clinic, okay? And if you have model metrics that are good, you're quite far away from that final goal. So we'll discuss that. I will discuss differences of model versus process. So if you train a model, that's one thing. But I was working at AstraZeneca, for example, in computational safety. And then the question is not only build me the best model and is it deep learning or which features do you use? The question is how do people actually use it afterwards? How do you make the process better to the extent you want to see drug discovery as a process? Because there's so many random jumps and personal decisions. It's questionable whether it's a process, but let's call it process for the time being. Then there's the question of tech push versus science pull. And there's really influences how you approach a problem. I will explain that in a few slides. Then there are the outer layers as well. And that's really layers in society. And these are incentives that people have in small companies, big companies, in society. And there's current trends in society as well. What's actually the value of science? To what extent do facts matter? To what extent does perception matter in stories, social media, and so on? And those outer layers even influence how we build models, right? Which method do we choose? Is that a purely scientific process? Yeah, if there's push from the outer layers of those circles, that influences how you validate models. Yeah, so there's a connection between those. And I want to put this sentence into your head. It's a sentence that you read every day if you spend too much time on LinkedIn. Our model achieves 93% or whatever it is, high number, performance on particular benchmark, outperforms SOTA, so state of the art, yeah. So you're familiar with that term. So it's basically, it's a model that as good as it currently gets. And revolutionizes drug discovery again, and so on. I just want to put that into your head right now. We'll come back to that in 10 minutes, because that statement does not translate to practice. Yeah? And I want to immunize you, if you wish, yeah? And to be careful to question yourself if you train a model. Does that actually translate to practice? Yeah? One thing is to publish. Another thing is to do something that's useful. Both things can overlap, but they don't necessarily overlap. Okay, I'll get back to that. Just want to mention that right now. Many of you are PhD students or just about to start your PhDs or just after the PhD. And so I just want to share what I think matters if you want to become a scientist. Do good science, do something good in the world as well. And I think those things here mattered really for me. It's partially personal development, partially it relates to science as well, yeah, and that is to go diverse. So I'm actually a chemist by training. I used to program when I was young, when I was 10 or 12, and that then morphed into machine learning later. And in drug discovery, for example, you can understand your chemistry, but what you influence is biology, pharmacology, medicine, and your tool is machine learning, right? And if you understand only one slice of that, you don't understand anything because you don't understand the bigger picture, okay? So I would encourage you to learn about aims, the data types we deal with, and so on as well, to understand what you deal with and what the tools are we have at our disposal in the big picture. Then what helped me is to work in diverse organizations. So I was working in academia for most of my life probably, but also in big pharma, and we started a couple of companies. And you learn something different in different disciplines or in different types of organizations. And resources are different, the people you work with are different, timelines are different, yeah? And you learn something very different. And you can easily jump from one area to the other. I think in the old times, I still remember my PhD supervisor telling me, when I finished my PhD, so Andreas, what do you want to specialize in? I didn't know what to say because I had no idea. Actually, everything was interesting and so on. And I realized I actually like to be general, yeah, to understand the big picture and not necessarily to understand how to optimize this particular type of model and so on in every detail, yeah, because then you can work with others and achieve that together. And that's my answer to that, so to go broad in science and organizations and also in cultures. We just moved to Abu Dhabi, so to the UAE. I think that's now the 7th country or so we live in, and people work differently, and how you work with people works differently as well. And so also different cultures can help you understand yourself better and understand different approaches, how to achieve science in a given environment. All of that is very valuable in my opinion. And don't be afraid to do things. So if you ask people what they regret when they die, or probably a bit before, what they regret in their lives, yeah, the question is, they regret what they did not do. Yeah? So it's not what they did and what went wrong, what they did not do. Yeah? So we're afraid of doing, so to speak. And so, and it's normal to fail, but failing is learning. It's basically the same thing. If you don't fail, you don't learn. It's the same thing. Yeah? So don't be afraid of that. So stretch yourself a bit, and failing is all right. So that's personally where I came from. Chemistry, then I learned biology. That actually came in my Erasmus year. I was in Dublin, so that's where I read textbooks in biology, because I realized it cannot be only chemistry, right? Because what we deal with is biology, the systems we want to influence biology, and machine learning are learning by doing, basically. And that's kind of the organizations we worked in. So I started, did my PhD in Cambridge, went to Novartis. Pharma companies have excellent postdoc programs, Genentech as well, AstraZeneca, Novartis, and so on. So even if you want to stay in academia long term, do your postdoc in a company that can really help, or machine learning company if you prefer. Yeah, because you see, you look at questions from a very different side. So I worked for Novartis, then went to Leiden, Netherlands a little bit, and then as a group leader back to Cambridge with 1 year at AstraZeneca as well. And while at Cambridge, we started companies. I will mention them on the next slide as well, to put things into practice. So after paper 200 or so, it's quite unsatisfactory to just publish. Yeah, you just write things and put it on archive or into a journal. Yeah, but it's not very satisfactory. Actually, I wanted to see things applied, yeah, to be put into practice. It's tedious. It's not easy, but that's the point. Yeah, to just write words, it doesn't really change anything. So we started companies as well. And these are the 3 companies to give an impression of what you can do with others, I should say. So I was always scientific co-founder. So I was never the CEO of any of the companies. So see what your role is. And I'm a scientist at heart, basically, and to give input into how to do things. Yeah, that's my natural role. So what is your role? Try to find that out, and then you can assemble a team of people to build something together with. And so the first company we started was Helix in 2014, so 10 years, 11 years old. For data-driven drug repurposing and rare diseases. It actually felt quite late to start a company in AI and drug discovery in 2014. So Tim, the CEO, came to my office and said, "Hey, Andreas, we should do data-driven drug repurposing for rare diseases." And I said, "Yeah, Tim, good idea, but I think it's too late for that." Yeah. But it's actually, if you work in a field, you think the world changes quickly. But if you are a scientist, you're in the first wave, okay? Probably the world changing takes 10 or 20 years longer, okay? So don't underestimate that difference in perception. So that company uses data, gene expression, text, and so on to repurpose drugs and combinations. Has phase 2 compounds now later this year. We also started Farm-Enable, much more chemical company, to do synthesis prediction. I think there was a lecture yesterday on that. synthesis prediction for protein-protein interactions, so more atypical chemistry, if you wish. It's a different type of company. So Helix wants to develop in-house assets. Farm-Enable is more on the fee-for-service side, although there are now in-house assets being developed as well. And Pangea Bio is the latest company. So that company uses compounds from plants that have been used historically in CNS diseases, depression, anxiety, and so on. Because then you have a phenotypic, as it's called, proof point that there's something in those plants that probably or possibly at least works. So you have on the human level, you have evidence, or at least historical evidence, you need to validate that the compound or the plant that people take has an effect in humans, okay? And then you can isolate compounds and you can put them, you can modify them, you can optimize them and so on, and can put them into in vitro and in vivo systems. Because the problem is in drug discovery phase 2, so you put compounds into the clinic, safety is more easily passed, even if your compound doesn't do anything, it's safe. But phase 2, your compound needs to achieve something in diseased patients to cure the diseased patients, right? That is much more difficult to achieve. Overall, probability of success 30% or so in that phase, so only 1 out of 3 makes that phase. And we are trying to increase that by using compounds from plants that have been historically used in the CNS area. And 84% of compounds that work in the CNS, so in the brain, actually have a scaffold, so natural chemical structure that resembles or is identical to that of plants as well. So these are 3 companies, most are in Europe, but one is also in Hub 71, it's a startup incubator in Abu Dhabi. So my point here is, if you do something, see what do I actually want to achieve with that? Is it only publication, or do I want to do something real with that? And it can be a long way, can take 10 years and so on, but have that in mind. You're one of the leading scientists, or many of the leading scientists here in this planet, right? If you don't change the world, then no one will, okay? So ask yourself what you want to achieve. So any statements I make here on my capacity as an academic, so it's not a company statement of any of the companies, and I would like to recommend this particular publication, Artificial Intelligence in Drug Discovery, What is Realistic, What are Illusions? We published that a few years ago, and we discuss what is actually different about data in the life sciences and other disciplines. A short version will be on the next slides as well, but in those papers that's discussed more explicitly. And it's understandable by people from computer science background, or pharmacology or chemistry background. It's written in quite generally understandable way, I believe. And that paper, I think, outlines the differences much more explicitly than I have time to go through today. So what's the big picture? So before we come to those circles, what's the big picture today? So it's not the first time that computers in drug discovery I actually put at the forefront of science. So the first wave, I think there was one before, but the first wave when I was on this planet was actually this one from 81, Fortune cover, proclaiming the next industrial revolution. So this is the year when Silicon Graphics was founded. Doc, so one of the docking softwares, was published the first time. It was very structurally driven. You could visualize proteins. You couldn't really do that before. It was difficult. You had to build models, plastic models. Yeah. But that was the first time when proteins could be visualized properly. You could rotate them and so on. That was the first wave. There was one in 2000 as well, when the human genome sequence was published, the draft version. There's still parts that get assembled today, plus there's biological variation in that anyway and so on. But there was another wave then around 2000. Bioinformatics became a household name. There was a biotech bubble. Yeah, that was 99, 2000. And these are some of the headlines we have today, how AI is changing drug discovery and so on. And so these headlines are from 2018 or so, but they didn't change much. We will look whether that's actually factually true, what is claimed on those headlines on the next slides as well, okay? The headlines don't sound so different. Certain things changed, others didn't. So when I was a postdoc at Novartis 2008, there was a data federation initiative, it was called, to actually annotate screening data with UniProt IDs, with protein identifiers. It wasn't done before. It was quite a mess, basically. The data looked horrible. And so the idea is that you can build, for example, machine learning models, or you can simply look up what you know, okay? And I think that's actually one of the bigger contributions of machine learning and drug discovery, that companies started to organize their data, yeah? To add identifiers that you actually know what you know. Yeah? That's already half the goal. And many machine learning models are not better than that anyway. You have a effectively nearest neighbor also classifier. Yeah? But to know what you know, to have standard identifiers is actually key. So that changed. We have GPUs, TPUs, and so on. That's all very powerful. What did not change though is the question, what's the early stage assay I need to run to have predictivity for the in vivo system? So what's my data point I need to generate in the test tube, if you wish, to tell me something about human efficacy and safety? That is still the question today, okay? So we have more data, but is more better? Not necessarily. We'll come back to that, okay? So the question is how to make a decision in even larger spaces. It even makes certain things more difficult. There's that saying, too much information is no information, yeah? So just generating data is not necessarily a solution. But let's come back to those headlines. So that's what those headlines look like. Is that supported by data? Not really. Here, that's from a Nature Reviews Drug Discovery paper, 2.5 or 3 years old. And they looked at what they called AI native companies as well as the big pharma companies. And they were looking at the stages in which projects are in those different companies. Discovery means basically anything up to animals. So you screen your compounds, you do chemistry, you put them into in vitro assays and so on, up to animals. You're into a discovery of preclinical. Phase 1 is the safety phase. So you have healthy volunteers, a small cohort, and you see, do I see adverse events in healthy volunteers? You can have some in healthy volunteers. Then phase 2 is small-scale efficacy, diseased patients. Do I see actually that my compound improves the situation in the disease? And phase 3 is large-scale efficacy, okay? And what you need to show that your compound actually does what it is meant to do is you need to pass phase 2, right? If your compound doesn't do anything, it's safe, you pass phase 1. But if your compound is meant to achieve what it's meant to do in the disease, then you need to pass phase 2. But you see, phase 2 is this one, and there's basically nothing up here. There's now 1 or 2 compounds that are in small-scale combined phase 2, phase 3 studies and so on. There's a little bit happening, but if you compare that to the headlines, how AI is changing drug discovery, you don't see that in overall approval rates. You don't see that in the pipelines of AI-first companies. So I think the clinical translation is still outstanding, okay? So don't believe all the headlines that you read. Look at the data, right? As a scientist, always see if the conclusion and the data actually match. And in those cases, I don't think that is really the case. That being said, docking, for example, so you have a protein structure, you're trying to dock ligands, has been used for 30 years or so in pharma companies. It's a staple. It contributes to every project. You can visualize how your ligand binds. You can decide how to optimize it better, and that does certainly have a positive impact on projects. But it's very difficult to quantify. Yeah? If you have a drug discovery project, you have 1,000 decisions. The computer makes 100 predictions along the way. So how can I actually say why that compound was picked? It's nearly impossible. Okay, but when it comes to approval rates or clinical phases, basically the proof point is still outstanding. But I think it needs to happen pretty soon because one of the promises was that AI and drug discovery makes those processes faster, right? And we had that now for 10 or so years. In big pharma, the distribution is quite different. You have many more projects in phase 2 and 3. And big pharma in-licenses compounds quite often, right? So they are clinical development machines, if you wish. Some people said that Pfizer is basically just a bank with a clinical development associated, because they in-license compounds, then they develop them. So you have a very different distribution of projects. The number contracted a bit because many companies focused on fewer projects recently. But the point is, clinical translation, we don't really see that yet to the extent it was promised. And that's what people realized. That's from a more recent Nature Medicine paper. So 2018, AI was already changing drug discovery. And now 5 years later, it's suddenly a nascent industry of AI-designed drugs. There's no shortcuts. So these are some of the CEOs of the AI-first startups that recently actually needed to close down or were acquired. So they said there's no shortcuts to drug discovery. The trials are still in the early days and so on, right? So it's easy to build a model, preclinical model. If you go into the clinic, you're confronted with complex biology, distribution of targets, biological variation, and so on. And that is where it gets difficult. Biology is difficult, not building a model. Have the translational aspect in mind when you build models. It's not the first time that happens. So 2000, I still remember the biotech bubble as well. That was the first bubble where I was alive that I consciously perceived. And also changed, I thought, with the internet and everything, the world will be a different place in 2 years. Well, it changed gradually, right? There was impact, but it took much longer and differently than expected in those times. And these were some of the expectations back then. So the human genome was sequenced in 2000, and 1 year later, This statement was made in a review, in this review. There will be more drug targets, 3,000 or 10,000 targets, compared with 483 at the time, okay? But 15 years later, or 16 years later, estimates of drug targets put the number at 667, so not 3,000 or 10,000, yeah? So it's easy to overestimate change, although there's a time element to that, right? Of course, some of those, we were able to catalog, for example, genes. Before there were expectations, humans have 200,000 or so genes. It sounds ridiculous in hindsight, although with the dark genome, maybe it's not as ridiculous, who knows? But at least one of the conclusions from the Human Genome Project is that we have 23,000 or so genes, right? And then you have different splice variants, and then you can phosphorylate or modify proteins, yeah? So it gets complex. But there are no 200,000 genes in the way genes were defined in those times. So it's easy to overestimate what will happen. And that interests me. How do we go from technology and potential to applications and better decisions? Yeah? So how do we translate that to practice? To come back to those circles here. So what I will start with is I will outline what's actually the goal in drug discovery, and then we will go through those circles from inside to outside, and I will outline what some of the problems is to achieve that goal. So what do we want? Safe and efficacious compounds in the clinic. So here, that's also from the Drug Discovery Today paper, we modeled if we are faster, better, cheaper, what actually has the biggest impact on drug discovery across the different phases, right? So in hit discovery, you do a screen, for example, you find your compound, you optimize that to a lead optimization, and then you can put it into animals, and then you have your safety, efficacy phase, and then your compound gets approved. And what you see is that the big bars represent net savings, and you can map all of those factors, being faster, better, and cheaper, on a money axis. Because if you're faster, you have longer patent life, right? If you're cheaper, you save money. And if you have higher success rates, you have fewer iterations of an optimization cycle. So you can map all of that on a money axis, and then you see the higher the better, and you see the highest bars on the clinical phases. And that means you have most impact if you have better compounds in the clinical phases, especially phase 2. Why is that? Success rates are low, 30% or so, averaged over different diseases. The phases take long, and they are quite costly, okay? And if you can address that, that has biggest impact, of course. Especially if you have better compounds in the clinic. What does it mean? The right patient, the right compound, and the right patient dosed in the right way. Yeah, that's what it means. But of course, the decisions you make are far before clinical phase 2. Because even before you start your project, you decide on a target. Even before you start screening, you decide on which chemical space you investigate and so on. So basically, the decisions you make even before the project starts, you only see them in clinical phase 2, right? So you have a 10-year or so lag between making a decision and understanding if something works or not. And that's one of the big problems of building models, machine learning models in drug discovery. You don't have a fast feedback loop. In the internet, if you do A/B testing, for example, you can change the button on your website, you increase click rates by 10%, you have the data in 5 days, and you know whether it works or not. Yeah? And so that's fast. 10 years is slow, right? You need to do something, change a process, which is another problem, and only 10 years later, and after spending $100 million or so, you know if something works, okay? And that lag is one of the problems to incorporate machine learning efficiently in drug discovery. You don't have a fast feedback cycle. Okay, but what matters is the clinic, okay? Especially safety and efficacy in clinical phases. That's what matters. So that's the goal, okay? So what are the problems when it comes to data? So the first circle of that is dealing with biological data. So machine learning needs labels, but biological data is impossible or very hard to label. Machine learning needs labels, so of course you have supervised methods, unsupervised methods, and so on, but at some stage in drug discovery you always need to make a decision. Is that series better or that one? Is that compound good enough or not? Yeah, so you always need to, at some stage you need to label. And the question is, can we actually label data in the biological space? That is very, very difficult. Why? I will illustrate that. Sometimes you see papers in machine learning journals that try to build a model and predict adverse reactions of compounds, or whether a compound will show an adverse reaction in a patient of a certain type. And then there's the question, does a drug cause adverse reaction Z? And then data gets used from MedDRA or CIDR or one of those side effect databases, and a binary classifier maybe gets trained. and then you have an AUC of 0.8 or whatever it is. You publish your paper, you think paper done, published in a good journal, great. But is that a useful model? It's not. Why it's not? So, because the data looks different, the data is much more complex. So, does a drug cause an adverse reaction? Yes or no? That is not a good question to ask, because the data has lots of other aspects to it. A drug might cause an adverse reaction if you have a patient with a certain genotype, with a certain disease endotype. Things depend on dose. And then we see an adverse reaction in a certain terminology. If you have adverse event databases in Japan and Europe, people have a different propensity to go to the doctor to report adverse events. There's different terminology, okay? So that terminology might be different. You see it in a fraction of cases. It might be a rare event. Then you see it with a different severity. How do you encode that? Maybe you see it only if you co-administer your drug with a certain other drug. Older people often take 7 or 10 drugs in parallel, so you have interactions. Maybe you see it only in males, maybe only long-term and so on, right? So it's very conditional data. So the question, does a drug cause an adverse reaction? It depends, right? It depends on at least those 10 or so factors, okay? So you cannot just label whether a drug causes a particular adverse reaction. Yeah? That is not a good way to represent the data. So does a drug cause an adverse event? It depends. For example, there's this paper here, death by water intoxication, okay? So if you drink water, 3 liters of water in half an hour, you dilute electrolytes and you have a 50% chance of dying. Yeah? And so is water now toxic? Would you label water as toxic? Obviously not, right? Things depend on dose, and that's true for any compound, okay? So how do you label your data? That is very tricky to do, and it's more tricky to do the more you go into the in vivo area, okay? If you have a biochemical assay, isolated protein assay, and so on, it's reasonably easy to label your data. If you go more into the in vivo data, vivo direction, and that really matters, the clinic matters, as I outlined before, yeah, then it's more difficult to label your data as we see here, okay? And that's different from many other areas. So let's come back to that statement. Our model achieves 93% performance. On that benchmark, outperforms SOTA and so on. Well, it does not really matter because drug discovery is not label prediction, yeah, because the labels that we have are not meaningful for the in vivo context that we eventually want to predict. I'm a bit too black and white here, I have to admit, because sometimes if you predict in vitro labels, you can anticipate something roughly and so on. But by and large, that is still true. Biological data is much more difficult to label than data in other areas. Yeah. And that means if I build then a model that achieves then better performance on a benchmark, that is then publishing a paper, that's one thing, right? But achieving something useful in practice, another. But of course, they might overlap. But my point is, try to build a model, try to validate it in a way that matters in practice. Okay, so the model is useful for the intended purpose. It might be used in industry. You might start your own company based on that, and it's not only a publication. That's my point here. Yeah. So that statement by itself, if the labels are not meaningful, is not a useful statement. Is the point here. And the image domain, for example, is very different from the drug discovery chemical domain or biological domain, and that's illustrated here. So if you have image classification or speech classification, you have convolutional networks, right? And you have recurrent networks, for example, or other architectures. And in the image domain, as an example, here you have an object, you have a cat in a picture, it's represented in matrix form, so you have 24 bits or so depth here. Here it's 1 bit, black and white, but it's the same principle. You can represent your image in a matrix. You have your convolutional net, for example, that operates on that matrix, so you can detect particular shapes, and in the later layers then maybe you have an eye that gets detected and so on, and you have 1 bit at the output that might then jump to 1 if there's a cat in the picture, for example. So you have a very good integration of object representation as a matrix, the model. So the matrix representation is a good representation of the object. Your CNN operates on the matrix, and you have largely unconditional labels. There's a cat in the picture or there's no cat in the picture. Of course, it can be partially hidden and so on, but compared to biology, that is a largely unconditional label. Very good integration of that pipeline. You can represent the object well, your model operates on that representation, and largely unconditional labels. And that's good for machine learning, right? So you can train a model based on that, if you have enough data points and so on. Very different situation in the chemistry and biological domain. Chemistry, for example, if you have a molecule that can rotate around bonds, you have different conformations, You can protonate your molecule. You can have different tautomers, so your protons jump to different places. There's different, not only representations, different states of the molecule, if you wish, different protonation states. You can also have different crystal forms. So not only one molecule, right? In practice, you have a powder or crystal of your molecule, and they interact. And you might not see that from the individual compound structure either, but that crystal can influence how quickly your molecule dissolves, which influences the concentration of the drug in your bloodstream, which influences whether you see a side effect. So there are links to all that. So this thing is alive, basically. There's not only one way how to represent your molecule. There's not only one way how to model that. There's lots of trial and error here, basically. And your label is conditional, okay? And that connection looks very different in the chemical domain, also the biological domain, by the way, even more so, I should say, compared to the situation here. In speech and images, you can label your data. In speech as well, you have a waveform, and you can say that part of the waveform corresponds to the word car. Yeah? You can say that. That's not really the case in the case of the chemical and biological domain. Okay, so that integration is much worse compared to the image and speech domain, and that's why also from the integrative aspect, many of the advances from the machine learning area don't really translate to the chemical and biological domain. Okay. And so I think the bottom line here is, especially if you start now your degree and so on, it's important to understand the problem that you aim to solve and the data available or which can be generated. And that is key to impactful machine learning models, right? To understand what do we try to solve, what are the labels we have, how good is that integration of the objects, representation, model architecture, and the labels, and the type of validation I choose for the models, of course, in the end. Yeah. And here, to give you some impressions of what data can look like in our area. So here are two plots from very different areas. In many cases, in the drug discovery domain, we operate in proxy space. We cannot just synthesize 1,000 drugs, put all of them into patients, right? So we need to have assays, so test tube experiments, to determine how well our drugs actually work. And what is shown here is, for example, for a drug-induced liver injury model. So the liver damage is one of the main side effects that drugs can have, because that's where the drugs get metabolized. And you can have what's called organoid models, so complex biology models that represent a small liver, if you wish, to test how toxic the molecules are. And what is plotted here on the x-axis is the activity in the organoid model, normalized to the maximum blood plasma concentration of that drug. So that is the activity. The lower, the more active, and the higher my concentration in the bloodstream, the smaller that ratio becomes, so the more negative my log becomes, right? So basically the left part of the axis here is the problematic one. If I have a very potent compound or a dose very high, then I'm in that region. And this is the clinical DILI liability in this particular model. And that's the in vivo liver damage, basically, that you see with those drugs. And here, the smaller, the worse it is. So that corner here is the most problematic one. But if you correlate our predictive model with the clinical model, ideally you would have to like to have a line like that, but you're far from that, right? So basically, and that's a typical example. I don't point that out because of that particular paper, but the point is that's quite a complex biology model. It's an organoid model. It's not easy to grow organoids and so on. But even that model does not really give us the damage of a particular compound in the human being, okay? It's not very predictive, is my point. And quite often in practice, if we are in drug discovery, quite often the data we have is not really predictive for the in vivo system, okay? There's a trade-off. How easily can we generate data, and how much does it actually tell us? Predictive validity, I will come back to that in a second. And on the right-hand side is another example. It's a thermal shift assay, as it's called. So it's basically, if you have a protein, you can measure at what temperature it denaturates. And if a ligand binds, then the idea is it stabilizes that protein, because it binds, and so the temperature where it denaturates increases. And or it might decrease in some cases as well, but it should actually increase if it stabilizes that protein. And you can correlate that thermal shift, as it's called, with the IC50 against the target. You do not expect correlation in this case anyway, because that's not a linear relationship. But you see here that you basically don't have any real trend. Yeah, so there's no thermal shift cutoff where you enrich the more active compounds. Okay, so it's a very low predictivity system as well. And there are many examples of those assays and the data we have that we need to deal with. Just modeling data and labels doesn't solve the problem. Okay, supports the previous point. So we model one axis, for example, this one. The one that matters is that one. Yeah, but the data we have isn't predictive for that part. Okay, same thing here. That's what we measure. What we're actually interested in is the IC50, but that axis is not predictive for the other one. And that predictive validity problem, that's actually one of the big problems we have in the life sciences. Now, we cannot put every compound into humans, so we need to have simple early stage assays that are easy to run fast, but those data are often not predictive for what we actually want to do. And that's another paper I can recommend. I will put the slides into Slack afterwards. So by Jack Scannell, so he was working on predictive validity. And what he's plotting here is, on the x-axis, my proxy system, in vitro potency, for example, test tube activity. And this is the data you measure in your assay and which you have available for selection. And this is clinical utility in patients. The higher, the more active, and the higher, the better. And the compounds you're actually interested in are the ones up here. But that's not the data you have. In practice, you measure only along that axis. So you can select only from the right. But what you see is, if you select from the right, you select some red compounds, but also many of the black ones, okay? So this system has a low predictive validity. If you have animal models, but many animal models don't work that well either. But if you have a better model, I think that's what it's meant to show here. If you have a better model, and you select here from the right, you only identify red points in the beginning, so good compounds, right? So this is an example of a model with a better predictive validity. And currently there's a trend towards large numbers. You have ultra-large chemical libraries, you have spatially resolved sequencing, single-cell sequencing, and so on, right? There's more and more data being generated, but the question is, first there's a signal in the data. That is the big question, and it gets more tricky because you have higher dimensional data and so on. But the underlying question is, which variable in my input data actually tells me something about the criterion that I'm interested in eventually. Better models is basically what we need. And that's what Jack was also modeling here. He asked the question, is it better to test more compounds in a screening system, for example, so to have higher numbers, or is it better to have a more predictive system? And so in those plots here, you have a predictive validity. It's in their case defined as a Pearson correlation between input and output, but you can define it in different ways. And on the y-axis, the number of candidates tested. And basically, if you start here, this white point, and those lines tell you how many of the compounds we're actually interested in do we find, okay? And if we start, for example, here at the white point, then testing 40 times more compounds is actually equivalent to increasing the Pearson correlation coefficient between my predictive variable and the one I'm interested in by 0.1, okay? So I need to screen 40 times more compounds, or I can improve my model performance, the correlation coefficient, from 0.4 to 0.5 or so. It has the same effect, okay? So basically, bigger numbers only bring you so far. Improving predictive validity can, where we're able to do that, can decrease cost, yeah, and allows us to make better selections. So don't think that higher numbers are always better. Often they are not. I want to have only one bit, basically, when I do drug discovery and not terabytes of data. I want to have one bit, and that bit jumps to 1 if I put the right compound into the right patient, dosed in the right way. I want 1 bit of information, not terabytes of data, because then you need to dig through it, and hypothesis-free data can be a real pain to go through. Yeah? There's a needle in the haystack problem. So it's actually sometimes less data is better than more data, but the right data, right? Predictive validity, okay, that's what matters. Yeah, that's what I just said here. That was a trend actually since Rutherford found the atom, or then genetic advances in biology. We could clone receptors a couple of decades ago, right? To actually to understand science or our environment by taking things apart, right? Understanding what atoms do, what is a receptor, signaling pathway, and so on. But certain effects are only emergent properties of a biological system. And if we then only look into the constituent compounds, that sometimes doesn't give us the full picture. Yeah? So we have lots of data. Yeah, but what we want is not more and more data, but actually we want data that contributes to decision making. Okay, that is the point I was just making. Yeah, too much information then can become no information otherwise. No. Okay, so that was the first part. So labeling life science data is very difficult. Okay, that is the first part. The second circle, how do we actually measure performance? So if you train machine learning models yourself, you're probably familiar with the area under the curve as a metric, or precision recall curves. So here, what we have is a recall plotted over relative rank. You can also plot precision recall curves, but here it's a recall plotted over the relative rank. And the question we ask the model, for example, is, I have 1,000 compounds, and I want to build a model for activity, and I ask the model to predict the most active compounds on top. Okay, so the model ranks all the compounds that I have according to predicted activity score. Let's say there are 10 active compounds in there. Those 10 would be at the top, the 990 inactives below that, and that would be a perfect model. Gives us the 10 actives at the top. So you would have, in this particular case, a recall of 1 in the first 10 positions, so somewhere up here, a relative rank of 0.01, so quite here at the beginning, and these are then the 1,000 positions of your ranked library. That would be a virtual screening setting. But the point is here, so here plot the recall over the relative rank, and you have two models here. So you have one model plotted in the blue curve, another one plotted in orange, and these are the curves to what extent and where in this ranked list they actually find active or toxic compounds that you might then select or deselect. And both of those curves have the same area under the curve. So area under the curve is one of the generic performance metrics that you might employ to compare models. Yeah, and the higher, in principle, the higher the area under the curve, the better. Because in my example, if I find the 10 compounds at the beginning of this type of curve, an area under the curve very close to 1. But reality is more difficult than that. So, and here we have two models that have two curves that have the same area under the curve, but one model actually has a 10% of this ranked set of compounds, has a double the recall. The orange one is double the recall of the blue model. And at the bottom of the list, the blue model actually has a much better false classification rate than the orange model, threefold or so at this particular position of the ranked list. So they have the same area under the curve, but they behave very differently in different areas of that curve. Okay, what is the point here? Generic performance metrics are not matched to the use case. Okay, and that is the problem. So in practice, area under the curve doesn't matter. In practice, what matters if you perform a virtual screen, for example, is how high is your recall at the top of the curve. Okay, and then this data point is more important. If you have a different type of model where you want to deselect, for example, this particular position of the ranked list is more important. Okay, and the problem is if you train your model and you have a generic performance measure that's not matched to the use case, it doesn't tell you anything about the performance of the model in a particular situation. AUC is tricky anyway because in practice you never use the AUC for anything. So if because you're making individual predictions about compounds. So your question is, I have this particular compound, what should I do with it? What is the clearance of that compound? Is it soluble and so on? Okay, and even if I then know the AUC of my model, it doesn't really help me with the decision making for that particular compound because AUC is a set metric, right? It's a metric over the whole data set. But what I need is RMSE, for example, is more useful in practice. I want to have a point-based error estimate, right? That tells me, in principle, how well can I trust the error of the prediction of that individual data point. But the point is, if you train a model without a use case in mind, you don't know which metric to use because there's no use case you have, right? And then you have a generic model metric which doesn't matter in the practical situation. So always put the use case first, is my point here. So this is one problem. What is the numerical metric we use for model evaluation? Another problem, and that's very typical to chemical space, is shown here. So chemical space is large, 10 to the 60 compounds or so. And what you have here is every galaxy is basically a chemical series. And in practice, what you have, if you have compounds and properties, let's say bioactivity or solubility and so on, you have a training set. So certain parts of chemistry that you have available for model training. You have your validation set, so you stop training at some stage, and then you have what you would call the external test set. But of course, in practice, because you very frequently split your data, all the data comes from, let's say, the same 100 or so publications. In practice, of course, the test set is hardly ever really external. External in practice would mean really prospective use case, a new project. But if you split your data, you draw all your data from the same underlying distribution, so you're lying effectively into your own pocket when you validate the model. That might help with the numbers, but it will not help in practice with the application of the project, of the model to the next project. Because if you then validate on an external test set, where you even have a bit of overlap with your training data, similar scaffolds, even if it's not the same compounds, but the next project will be somewhere else, okay? So what does performance on this area of chemical space tell you about performance in different areas of chemical space? Nothing, okay? So that's one problem with the metrics. What is the right metric to use in the absence of use case? The other one is the next project by definition is a new project, right? We don't want to predict yesterday's weather, right? We predict the weather next week or so. And the numbers we obtain on this test set doesn't tell us anything about the performance elsewhere. And that's the problem with many of the chemical models we have. In principle, you can have applicability domain measures and so on, but honestly, I've never seen anything that works. And one of the reasons is probably chemical space is large. We have very little data, and what matters is the absolute position in chemical space. Yeah? And if I don't know anything about the absolute behavior, absolute properties in one area of chemical space, I cannot infer that from another galaxy. Yeah? I don't know what the atmosphere on a planet is in a different galaxy if I only know our solar system. Yeah? I don't know that. It's different. It's just different. Okay, and that's different from many other areas. So we cannot really validate our models since our models operate mostly in proxy space and in a retrospective manner. And that's different from tech. In tech, you have annual recurring revenue, if it's more commercial, user growth, click rates, precision recall on somewhat meaningful labels. If you do bio-ML, you don't really have any of that. You always operate in proxy space, okay? And you might get good numbers there, but it's then translation to the in vivo situation that really matters. That is the tricky part. Okay, so that was circle 2. First one was labels. The second one, the metrics, and do they hold for future applications of the model. The third one is model and process. So if you are a machine learning person, you might focus on those parts here. So you have data, you put them into a model, you do a cross-validation, external test set and all that. And then you get a prediction confidence, very good AUC, let's say, or average precision or something like that. Publish your paper and you're done. And so you improve model performance. But is that what the real world looks like? No. And because in the real world, you don't only have a model, you want to impact a process, okay? And that process has a compound with a project context, and that might be a lifestyle disease, it might be late-stage cancer, it might be a compound dosed in huge doses, it might be one that's dosed at a very low concentration, and so on. It might be a compound that has a different distribution in target organs, and so on. So there's a context how you use the model. And the other part is you have follow-up assays. So my model predicts something, what next, right? Because a prediction is not the end of the story. The question is, how does that impact the real world? And both of those, if you ignore both of those, then your model can actually not really contribute to the process. Why is that? So if you have a model and you build, let's say, a binary classification model at a given activity value, 10 micromolar, active, inactive, okay? But then your project context, you have a compound that's dosed at a 10 nanomolar level, let's say, right? Can you use your model? Of course not, because it's generated at the wrong threshold, right? And so on. There are many problems like that. In this regard, if you have the model that you train and validate only within this circle, but you don't take the use case into account, also links to the performance metric part, of course. If you don't take the use case into account, you cannot build a model that you can effectively use in future situations. You need to approximate that use case as much as you can. Just building a model is not enough. The other part is follow-up assays. My model predicts the compound is 83% likely to be toxic, but you spent already 10 million on that compound. So because the computer now says, I'm pretty certain that compound is toxic, you now scrap the project after spending 10 million. That's quite unlikely, right? So the question is, my model predicts something, what do you do next? And that's the question, what are the follow-up essays I can run? What can I run tomorrow? An essay that's predictive enough to tell me something about is that really true or not? Yeah, that's the other question. And I've really seen that at AstraZeneca. So we were running or building models for the in silico safety group. And the question was partially, of course, do you have good models? But the other question is, do people use them? The model predicts something, what should I do? If you cannot do any essay, you create more uncertainty, okay? You want answers and not more questions from the models, right? So the question is, what are the essays you can run afterwards? So basically, that's the model. If you only care about the model, that's insufficient because it is embedded in a process, and to understand those two interfaces is key if you want to impact reality as well with that model. Model metrics by themselves are insufficient, right? So it's about process impact. And that also has cultural components. How do you educate an organization of 10,000 people that our models are actually pretty good and they should be used, right? If people don't trust them and maybe on their first 3 compounds, they did not work, right? These are the questions that matter in practice, yeah? Not necessarily how deep your network is and so on, right? So care about the process, translation to reality, right? So to come back to that statement, so our model achieves that performance and so on. So it often doesn't really matter because it's a model metric, right, and not a process metric. So the question is not only in my publication, how good is my model? The question is, if I use it, can I come to better conclusions, better decisions, right? And of course, in papers, you wrote often have that. Circle 4, so tech push versus science pull. So quite often we are right now in a situation where the tech hammer looks for the use case nailed, so to speak. So tech push inverts the logic of purpose, data, method, and how you use the eventual model. So that's actually something, Koichi was a visitor from Japan in my group a few years ago. We still write papers right now. Actually, it just came out in Drug Discovery Today a few days ago. So if you're in science pull, you put the question first. I want to solve a problem. What's the right way to solve it? Yeah. So you have a project purpose, and then you want to, for example, I want to stratify patients in this and that cancer. How do I do that? Well, they overexpress this and that receptor, so I need to do RT-PCR or gene expression profiling to generate that data. Then I have a method, machine learning method or classifier of some sort, and then I use that model in practice, okay? And then the purpose, the question, the problem I have that I want to solve influences which data I generate. So I generate the right data to address that question, then I use the right method, and then I have the right model in the end. But tech push, and quite often right now, tech push happens everywhere, that inverts that logic. So actually it starts with the data, and let's select all data. Sequencing is cheap, so we look into sequencing data, yeah? Or we already have that data set, so that's now what we use for model generation, okay? So we take whatever we have, and then we use a method that might then be the flavor of the day, as some people say. So now it's deep learning, because that's one of the methods of interest. I evaluated the model using generic performance metrics. Then I have a model of some sort that I'm trying to apply somewhere, okay? That inverts the logic, and then you don't really have the right data, the right method, with the right evaluation for a particular use case, right? So basically, always try to distinguish between science pull and tech push. So in science pull, you're trying to come up with a solution that aligns the question, data, method, and the use. And tech push that can be inverted and it not lead to optimal results in many cases. You don't really have the right method, evaluate it in the right way for the right purpose. And that's a kind of comic that Koichi came up with. He's a DMPK, so like ADME, so compound property, drug metabolism and pharmacokinetic scientist. So how does a compound get metabolized and excreted and so on. And he came up with a hypothetical dialogue between that scientist and a computer scientist. And I think it's really, I mean, the dialogue didn't happen, but it's, I think it's really an illustration of what could happen. So the scientist says, oh, MedChem has been asking for all the compound data. It's kind of quite a pain. I wish AI could handle that. I use ChatGPT, but I don't know how it works. So I cannot really do that myself, build those AI models. And the computer scientist says, oh, wow, I didn't know there was so much ADME data published. Numbers is numbers. Let's just plug them into a model, right? I could write many papers, change drug discovery, right, with a fancy machine learning model. Okay, that's phase one. Then 6 months later, what does the scientist say? Oh, I heard from my boss there's an ADME prediction model on the website, on GitHub or somewhere else. I could check it out. But actually, they did strange things. The ADME evaluations were all over the place, predicting things that aren't typically used. It wasn't that helpful. So probably I have to do my experiments after all. Okay, so the scientist doesn't really get the model validated in the right way to be useful in practice. And a computer scientist says, well, I've released my model here. The machine learning researcher said that's a great model, but it didn't hear about it being used for some reason. Maybe the MPK scientists aren't interested in AI. It could actually reduce the number of experiments and improve drug discovery and so on, right? And this is, so what's the point here? The point is not that one is right and the other one is wrong. The point is to work together, yeah, to understand the use case. So basically, if you come from the computer science side, try to understand the data and the problem, okay, so you actually solve something that's worth solving in the right way. And if you're an experimentalist, then you communicate what is needed to the computer scientist or machine learning person. Yeah, so in the end, there's actually a model that's fit for purpose. That's the point, right? No. And that was the fourth circle. The fifth one is about incentives. And so incentives really drive what people do, right? In some countries, there was quite a focus on publishing in journals with a high impact factor, right? And then you get lots of MDPI publications and all that. So incentives really drive what people do, right? That's very important. So what happens actually in different organizations here? In our area, machine learning and drug discovery, so there's no real feedback loop on the long-term reward functions or clinical success. So you need to optimize on proxies. Huh? And that can look like in pharma, we need a winner, for example. So we generated terabytes of data, right? So just about generating more data, or we work with a particular company. So that's then the incentive that comes from management. In academia, we published another high impact paper and improved SOTA again, so on entirely irrelevant benchmarks. If you're in a startup company, what can happen is you're stuck in the pain of platform validation and pilot studies for quite a while. And platform validation is important, but my learning is do that to the extent you need to, and then develop in-house assets. That's really what the point is in our company. And of course, in grant funding agencies and publishers, we have similar problems. It's quite a vicious circle of we fund excellent research, but from the side of funding agencies, but actually that often means overhyped science of the day, single cell sequencing or whatever it is right now, published in high impact journals, right? And the journals publish what gets cited, so which is then the overhyped science of the day by people who've done it before, right? So incentives can actually lead to outcomes that are suboptimal in the big picture then. And that influences how people build models then as well, right? No. And that then leads to lots of overhyped pseudo-innovation. For example, you might have heard of some of the foundation models, genetic foundation models recently. And there was, for example, SCGPT, but there are 10 or whatever biological foundation models around right now. But there are now studies to try to evaluate, are we really better than some baseline models? And quite often that's unfortunately not the case. And that's one example here from genetic foundation models. Where it was found even the simplest baseline model, taking the mean of training examples, so it's really trivial, the most trivial thing you can do, outperformed SCGPT and SC Foundation. That's one example. Another one is from materials discovery. So there was this February when there was this Google DeepMind paper, millions of new materials discovered with deep learning. But 3 months later or so, there were follow-up articles in Nature that actually questioned whether any new materials were discovered. There were problems with the analytical chemistry components of that paper, for example. I quite like this quote here, which I don't know if it was meant to sound like a compliment or so. I don't think the entire work is garbage, says Schoop, but the analysis of the product clearly failed completely. So the question is always, how do you have machine learning models? How do you validate them and put them into practice, okay? It doesn't always work. And that is a problem in companies as well, because every year you have a new technology, what you can do, but you cannot evaluate it completely, right? So there's always a problem, how do you actually find out what works best in your use case? That also, and one problem with that is this so-called bullshit asymmetry principle. So it takes 10 times more energy to refute bad science than to create it. And that means that many of those claims actually linger on and on in the end. Yeah, and that is, I will go through the Google co-scientists as well. It's not specifically about Google, I should say. It's about many of those co-scientist approaches that are out there right now to go through the validation of those approaches and to show how novel are they actually, how valuable are those validations. So that was a February paper, Accelerating Scientific Breakthroughs with an AI co-scientist. And so it was an agentic model built with Gemini, and to generate novel hypotheses and research proposals, and that was the aim or claim at least, to accelerate the clock speed of scientific and biomedical discoveries. And they came up with a hypothesis, where they then performed experiments to validate this, in this case, Google co-scientists. But let's look at that. So how novel was that? How valuable was that actually? So the first claim was that the authors did drug repurposing for AML. And there's certainly disease that's in need of new treatments. So in principle, nothing wrong with that. But what they tested was a kinase inhibitor that was well known, that was found to have a pretty good activity in AML, so in AML leukemia cell lines, right? That was the test that they performed. And there are two big problems with that. The first one is the claim is about drug repurposing. Drug repurposing, by definition, is in vivo, okay? You cannot repurpose a drug against a cell line, okay? That is the first problem with that. The other part is this compound is a well-known kinase inhibitor. You could probably put that on nearly any cell line and it would kill that cell line, okay? At least the ones that express the target kinase. And so it has very well established and very related activities in anti-cancer space already. So it's only a very, very slight extrapolation. If you give that to any human medicinal chemist, he would come up with that option within 1 minute or so, and very likely it would be right, okay? So the problem is this drug repurposing claim, that's overclaim in my opinion, because drug repurposing is something you do in vivo. And from the compound activity side, the extrapolation was very slight as well. If you look into PubChem, you actually find hundreds of data points of that compound being cytotoxic. The second validation or claim was that they advanced target discovery for liver fibrosis. So there was the claim of discovery of novel targets relevant to this particular disease. Again, laudable aim in principle, but the promise with the validation here was that no details of the discovered targets were given, so novelty and so on cannot even be assessed. And in the validation, there were plots given without biological detail. Drug effects, so compound effects on fibroblast activity were shown. Okay, so it's not a genetic intervention, it's a compound intervention, which is not entirely selective, yeah, and it's on fibroblasts. Yeah, so it's not necessarily target that translates to in vivo activity either. Okay, so there are some holes in the second validation here as well. And the third one was explaining mechanisms of antimicrobial resistance. But what the researchers did here is they discovered something in experiment and then asked the Google co-scientist whether the co-scientist comes up with the same thing as well. Okay, so whether it's able to rediscover what they just found experimentally. But that is also not what prospective discovery looks like. Okay, so be careful with the validation and the integration of machine learning models and evaluate what they are claimed to do and to what extent that is validated. And the biggest trend, of course, is trends in society. So there's now quite a transition of authority from experts and facts to influencers and public opinion and belief, right? And that is, and quite often is really difficult to follow some of the statements that people make. So that's a statement by Sam Altman from 3 weeks ago or so, from OpenAI. So he said on his blog, the takeoff has started, humanity is close to building digital superintelligence. It's much less weird than it seems like it should be. So I'm not quite sure what to make of that. So that's what it looked like when I read that probably. So there's the question is really to argue on the technical factual level to find out which methods work to validate it properly, right? In practice, you have quite a lot of money push. You see that on LinkedIn these days as well. So if you build models yourself, yeah, you always have problems cleaning your data. You don't get out of domain extrapolation and so on. That's what your real life looks like, right? But if you read LinkedIn, you have more the message, AI can do everything and will change the world. Well, in some aspects, maybe it will, right? If I have a good model that's predictive and so on. But still, there are huge overclaims, right, on what LLMs can do and so on. And that actually leads to adoption of immature technology. There are some examples from Klarna and Duolingo, So they try to replace some of the employees with machine learning models, but it didn't quite work, so they needed to rehire the people they kicked out just 6 months before as well. And on the pharma level, there's quite a lot of pressure on pharma to innovate from the stock market and so on, the owners. But in the absence of ability to validate, when it comes to the in vivo endpoint, it's often, of course, at best then pseudo-innovation. You try what you can do, right? But it takes 10 years to see what really works. So it's difficult to really identify which machine learning models help me best. And that gets exploited then often by tech-first companies. We know how to do this. We deal with how to deal with big data and so on. But often that's not really the case when it comes to drug discovery, as I've shown with the validation of the Google co-scientists, for example. Okay, but I think I will circulate the slides afterwards. There are a few examples where I believe machine learning models can help, but actually I would like to leave some space for questions as well. If there's time afterwards, I can go through some of those as well. Yeah, any questions from your side? Oh, please have some dialogue as well. Please. I think with the mic, I think I meant to use the mic.
Hello. So first, thank you very much for such insightful talk. So it's resonated with a lot of things I personally met in pharma places. And just a small question, you have one slide on comparing representations of images and molecules, one to the other. And you were kind of saying that for images it's much more clear and established and it's a good representation, but for molecules it's much more unclear, not defined, there are lots of variabilities. Maybe it's not a question, I just want to mention, so I'm coming originally from computer vision field. 12 or 15 years ago, it was also for computer vision error on trial. People didn't know which representations to take. There were very different types of representations and they didn't work so well. And also then recognizing cat or dog, it was also extremely complex task. Once it's found with CNNs and deep neural networks, now it seems to be trivial. But now what I want to say now is just also for chemicals and for molecules is just not yet found probably the proper representations. These mass, maybe they are not so proper representations, and what is absent, I think, is just amount of people that have expertise in chemistry and machine learning to come up with some novel types of representations for molecules that are more meaningful, and then it will also give a push. It's not the originally that this problem is hard, and with images it's easier. In images it was also very hard and with lots of parameters, and just in time, dozens of years until people came to very effective representations.
Yeah, yeah. I think, I hope you're right, and that changes. There are two aspects, though, or two reasons why at least currently I'm not too optimistic about that, but I might be wrong, of course. Let's see. The one is the nature of chemical space and the very local behavior. So if you have chemistry from a different scaffold, let's say, it's a different galaxy. So you can, in bioactivity space, for example, you cannot really extrapolate because you don't know which type of bioactivities you find in that area of chemical space. Yeah. So that is a high dimensional space of a very local nature, and that is one problem, I think. And the other problem is that is when it comes to bioactivities against targets. The other one is when it comes to in vivo extrapolation, the conditionality of the data for the in vivo case. It's kind of two different situations. Yeah? One is the local nature of chemical space, even when it comes to the in vitro situation, so where I can label my data, there the chemistry is the problem, so to speak. And the other part, and that is, I think, fundamental, is the conditionality of data in vivo space. We simply do not know if you give that patient 3 times the dose, what's the genetics of that patient? That is what matters. We don't know it. It's not in the variables. It's not in the system. And I think that's a systematic problem. That would be, yeah, let's see. But that would be very difficult, I think, to address. Huh?
Yeah, with the second, I completely agree. It's in vivo and in vitro, it's different worlds. Maybe you can't extrapolate. But with the first locality, also in image space and language space, it's also many things are local. You don't see correlations, but somehow there are methods that can tackle this problem. But thanks, thanks for it.
Sure.
Good morning. My name is Anna Krawajczyk. Thank you very much for a great talk. I was wondering, because that's true that chemistry is very, very diverse, and prospective finding actives in the chemistry space, it's difficult. But what do you think? Is it possible to change a little bit the paradigm, instead of looking at the compounds, looking, for example, at the proteins and the pockets? Because, for example, I have seen talk for Charlotte Dean, who was saying that most of our models are still very much rely on the chemicals, so on the compounds. That's why they are difficult to predict a different chemical matters that is active on the proteins. Are we able to find a different way of building the models?
Yeah, so of course what I talked about was quite specific to small molecule bioactivity. There are many ways how you can influence biology. You can have knockouts, you can degrade your protein, you can do many other things. And you don't necessarily need a small molecule. There are many other modalities around, antibodies or so, yeah. And sequence-based design, because you have a more limited alphabet of amino acids. AlphaFold, yeah, you have a much more limited alphabet of amino acids to predict antibody binding. I would bet my money on material science and antibodies instead of small molecules. Yeah, if I had to bet on one company, because the space, the underlying space of what I'm trying to model is easier to model. Yeah, so yeah, we don't need to have small molecules. If we can stay in protein space and develop an antibody that's efficacious, it's not just a binder, it needs to be efficacious, it needs to translate to clinical efficacy, then that might be a solution as well. And then you can circumvent the small molecule problems, many of which I outlined. That being said, in order to get into the cell, for example, certain targets, you need small molecules are also cheaper. They get into cells. They have many other advantages. They're getting into the brain, at least if you have the right small molecule. So there are some other properties that are difficult to achieve with some other modalities. In some areas, we might be able to circumvent the small molecule problems I outlined. That's right. That's right, yeah. And we can do that with proteins, mini proteins. There's so many other modalities around, yeah. These are all options, that's right. No. Yeah, usually what you need to look at, I think, is the overall profile. So it's not only on target activity, right? You need to consider PK off targets and what actually modifies disease, right? What translates to downstream effects. And based on that overall profile, I think you would need to choose which modalities can be relevant. But if your target can only be accessed by small molecules, it will not work in this way. And then we need to solve some of these small molecule problems. Yeah. It's about the overall profile. Yeah.
Okay, so I have more like a philosophical question. Do you believe that, because you said that biology is very complex, but do you believe that we can at some point decompose it into much simpler problems for which we can generate some reliable data? Do you think it's possible in some future?
Now, let's see. So, for example, you have in biology, you have stochastic processes, okay? You have very small copy numbers. You have a single copy of a protein in a cell. How do you know in a patient you dose tomorrow if in a particular cell he has that protein or not? Yeah? So there are systematic problems with that. Another one is on which layers can we actually generate data? We can sequence quite easily, but imaging live cells, if you have a patient and you want to investigate that patient's brain, how do you do that? Yeah, it's much more limited. Of course, you can do liver slices of animals that are dead, okay? But the biological systems you want to treat, quite often you cannot generate the data you would be interested in, yeah? Even if technology would be perfect, so to speak, yeah? Even if we could do, on a single cell, we can determine all molecules in there, all interactions and so on, which is quite far away as well. We only see slices right now. We can sequence, we have expression levels, yeah. But when it comes to phosphorylation states, proteome-wide phosphoproteomics, for example, yeah, and any other levels of biology that we're interested in, localization of any of those proteins, yeah, is technically difficult to measure. And in the in vivo system we are interested in, that patient's brain, that patient's liver at that time point with all the heterogeneity, I think there are also systematic problems with that. Similar to the previous question, there's one part that is partially addressable maybe by tech, yeah, but the other part is intrinsic to biology, and that is very difficult to address in my opinion. Yeah? And then if you generate data, you have high dimensional data again, time resolved, spatially resolved. Yeah, it's a very high dimensional variable system. You have many columns, few rows, yeah, because it's for that individual patient. And then you run into problems, how do I make a decision based on that data? Yeah? In my opinion, if you have gene panels, for example, for cancer, usually it's only a few dozen genes. Yeah? We know only what to do for a few dozen genes or so, how to, which drug to prescribe for a given cancer. Yeah? It's not a huge number. We don't understand much of that biology, which makes it not actionable. So we can generate data, but we cannot draw conclusions, practically conclusions from that, what to do next. Yeah? Of course, we move into that direction, but there's quite a lot of way to go, I think. Yeah.
Okay, thanks. So I guess the question is rather not.
Rather not what?
We won't be able to decompose the biology properly.
Yeah, that's what people try right now,
Yeah,
in many different ways, right? You can do cellular imaging, live cell imaging, let's say, yeah? But the question is, every patient is different, every time point is different, biology is heterogeneous, there are multiple levels of biology which you would need to analyze, yeah? Genes, proteins, and so on, localization, phosphorylation states, and you don't have those variables for the future patient. In some areas, yes, we'll be able to find, is it overexpression of that gene that gives rise to that cancer and that patient cohort? So where we're able to zoom in into few variables that generalize, okay? In those areas, the biomarkers, that's basically a biomarker case. Yeah? Where we're able to discover predictive or prognostic biomarkers, there, yes, but it's a minority right now, due to many reasons. Determining data on all of those levels, having enough sample size, yeah, and to be able to then stratify patients. To an extent, biomarkers work. Where they work, it's great, but it's a minority of cases where we have biomarkers. Yeah?
Okay, thanks.
So I also have a question. So during your lecture, you mentioned a lot of problems with translating the AI methods and getting state-of-the-art results to actually applying it to drug design, to the point where I started questioning whether what we do in academia is actually useful at all for application in drug design. So I would like to ask you to maybe comment on that. And also maybe do a sort of wrap-up for young scientists to tell them what they can do to make their research more meaningful and more impactful for the real drug design process.
Mm-hmm. I think there are areas where academic research can contribute. If that research addresses something, that's the applied angle. I'm quite a applied translational person. I mean, I don't judge that other research that cannot be applied is not useful. I mean, but from the applied angle. And I think that is, why do I do something conceptual, or do I show in practice in a relevant system that it works? Let's say you can, in Cambridge we did that as well in IPF, pulmonary fibrosis. We want, there's an 81 to 82, it's a transition between cell states. Yeah? And so we were developing an organoid model, which was quite a pain, I have to say. I'm not quite sure I would do that again. So an organoid model for pulmonary fibrosis, so biologically relevant enough system. And we were putting in compounds selected using gene expression data to see if we see that transition. And I would argue, of course I was involved, so maybe I'm biased here, but I would argue that biological system is good enough to be useful. It's a disease-relevant system. And so if we understand compounds that influence that transition in IPF, that would be practically useful. So to test things in a relevant enough system. Yeah, but even if you can only do in vitro tests, because projects are always limited in time and money and so on. Yeah, even if you test only activity against targets, it's better than nothing and only retrospective validation. So try to validate, try to go into experiments as much as you can. I needed to learn that as well. I was doing cheminformatics in my PhD, only modeling and retrospective validation. Yeah, and I thought that's good enough, but at some stage I realized, well, actually that doesn't really work in practice. So is it useful? And to keep that in mind, yeah, what do you really do with that? Yeah. But the second question was about students, what to take home?
So a take-home message, like what they can do themselves to make their research more meaningful and to do it in such a way that the results would be easier to translate to drug design.
Yeah, I think if your students, I mean, by definition, you're in academia, I would recommend see if your supervisor knows people in companies and industry. So someone who can give real world feedback on a particular project. Because that is, or your supervisor might have that angle as well, but sometimes in academia that viewpoint is not known because people didn't spend time in industry and so on. So if there's someone from a pharma company who could give input on the respective projects as well, yeah, to get that real, it also helps building a network and so on, and then moving towards a more relevant validation, yeah. So to get input from applied scientists as well, that probably would help, yeah.
Okay, thank you very much. I'm not sure if we have, yes, we maybe have time for a very short question, if there is one final question. Oh, there is one.
This is not so much scientific question, but you have mentioned on your slide in this circle that this is also like a matter of influencing the process of the drug discovery. People who are sitting in the drug discovery already 20, 30 years, and they have already their mind, they have already their experience. They do the things always like this, and sometimes it works, sometimes not. But do you have any advice, or maybe you can share your thinking, how to change this process, how to make the people going out of their boxes and maybe try the models which, okay, sometimes they work, sometimes they not, sometimes they help because they are in the proper galaxy, sometimes you need to test them and challenge and go to the different galaxy. So the question is how to influence the process.
Yeah. And in some of the companies where I worked before, I was also quite intensely involved in that. I think there are 3 or so aspects that help. Validation, explanation, and having good interfaces. What I mean by that is it's important to be credible to the scientists. Yeah? To the there's a history, of course, to computer-aided drug discovery. Right? Many people used the first version of some algorithm before, it didn't work, and that was the end of the story. Right? And I think as a computational modeler, you need to pay attention to what matters in practice, validate your model properly, yeah, and do it with experimentalists as well. So to validate to the extent you can validate, try to come up with applicability domains as well, putting out the nearest neighbor that's present in the database, yeah, those things. That's also a very active compound against that kinase, makes it more credible, so you can help a bit with that, so provide actual underlying data. So that is validation, interfaces. So interfaces, that's one aspect I just mentioned as well. We were also building, for example, at Astra, microsomal stability models. So does my compound get metabolized very fast in cells, microsomal cells? And here the question was, which type of model do we actually use? And because it's very noisy data, we chose then to build a categorical model. 4 categories, by the way, yeah, because if you have 3 categories, you predict the middle, you never quite know where you are. Yeah? So it was 4 categories in this particular case. Correlation model wasn't good because the experimental error was very high. And then the experimentalist, if he says your model gives a wrong prediction, that might be as good as the experimental error. But of course, the perception is that it's a model error. Yeah? So we built a categorical model. So interfaces matter. So first thing was validating interfaces and explaining it again and again, I think. Yeah? Changing culture is a very long-term process, yeah? And to really explain again and again how the model works, where to use it, and where not to use it as well, and to be honest with that. Yeah? That's the 3 things I think I came up with. And that's a very long process. Yeah. And that is explaining is the best approach I could come up with. Explaining again, and validation interfaces, and explaining again and again. It's a long road to change culture, right? I mean, society as well as an organization now. Yeah.
Okay, thank you very much for your presentation. So let's thank the speaker.
Thank you.